أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم السلام عليكم ورحمة الله تعالى وبركاته. Today I am going to talk about antigen recognition molecules. As you know, we have two kinds of antigen recognition molecules, including immunoglobulin or IgG or antibodies and T cell antigen receptor or called TCR. مثل ما تذكرون احنا ذكرنا بالمحاضرتين السابقتين general information about immunology and immune system and immune cells. Then we describe the antigen in general. Now I'm going to talk about the recognition molecules, including immunoglobulin and TCR receptor. لو سألنا سؤال, we have billions of antigens like microbes, other particles, but we have a few kinds of immunoglobulin, just 5 kinds of immunoglobulin and TCR receptor. And as you know, the main role we have antigen should have a specific antibody for that antigen. الآن راح نسأل هنا لو كان عندنا مليون من الانتيجينز كيف راح يصير لهم recognition من خلال 5 kinds of antibody. The main features, the diversity and heterogeneity are characteristic feature of these molecules. فراح نتطرق للdiversity وراح نعرف شلون يصير لهم recognition, specific recognition for each antigen. In both cases, there is evidence of extensive gene rearrangement which generate immunoglobulin or TCR capable to recognizing many different antigens. Then we should know the diversity, heterogeneity, and gene rearrangement. The first recognition molecule is called immunoglobulin or antibody. The antibodies, they are a group of a glycoprotein present in the serum or tissue fluids of all mammalian, including human. They are produced by B lymphocyte, or called the plasma cells. The main function or action of immunoglobulin, they act as a critical part of immune response by specifically recognizing and binding to particular antigens such as bacteria, viruses, and aiding in their destruction. As you know, the primary function of immune system is recognizing the antigen and then minimize the damage, as I said in the first lecture of immunology. What is the structure of immunoglobulin or antibodies? We have 5 kinds of immunoglobulin like IgG, IgM, IgA, IgD, and IgE. Each antibody has 2 functional regions. The Fab portion, that's the Y shape, that's the structure of antibody, the Fab portion, and Fc regions. The basic unit of IgM monomers consists of 4 polypeptide chains, 2 heavy and 2 light chains held together by disulfide bonds. The light chains, there are 2 antigenic types, which is called GABA and lambda. Only 1 type of light chain is found in any individual molecules, but we have 5 main types of heavy chains: gamma, alpha, and mu, and IgA, delta, and epsilon, the IgA. Each chain consists of 2 parts: N-terminal part, which is called variable region, and C-terminal part, which is called constant region. The constant portion of heavy chains also can be divided into 3 regions, CH1, CH2, and CH3. These globular regions stabilized by intra-chain disulfide bond and refers to domains. إذا كان عندنا antigen and the antibody should bind with the Fab portion وهناك كانوا ممكن المكان مالتهم ما متناسق. Then we have a hind region as a segment. This is the part of hind region. This is a segment of heavy chain between CH1 and CH2. The main function of it, well, flexibility is a flexible region. The main function is a flexibility in this area permanent to antigen binding site to operate independently وبالتالي راح يسمح بحركة الفاب portion. ايضا we have a J joining region, IgA and IgM, because we IgM and IgG have more than one monomers, dimonomers and pentamonomers. IgM and IgA have additional peptide chain, J joining chain, thought to assist the process of polymerization. بالتالي راح يصير عندنا polymerization بالالفا وبالميو. This table shows the main differences between Ig groups, Ig immunoglobulin groups. The main properties, in, as you said, in the body weight, the major body weight was the IgM 900, and then IgA, the half-life, the IgM, the IgG, sorry, أكثر half-life, and the molecular form with IgM pentamer, with IgA monomer or dimer, and other monomer. The subclasses of them, IgG has 4 classes, while IgM and IgA have 2 classes. The others have 1 classes. At chain, as you said, gamma, delta, epsilon, and J joint only with IgM, IgA because they have polymerizations, and cross placenta with IgG only, opsonization with IgG only, allergy with IgE only. That's the main differences as you know in this table. The interaction of antibodies with antigens. The binding of antigen to antibody involves the formation of multiple non-covalent bonds between the IgG and and antigen. I mean, between antigen and amino acid of binding site. The attractive forces, we have 3 kinds of attractive forces: hydrogen and electrostatic bonds, van der Waals, and hydrophobic forces. They are weak by comparison with other covalent bonds. We have 3 main terms: antibody affinity and antibody avidity and cross reactivity. We should know all of these terms. What is the antibody affinity? Affinity refers to the strength of a single antibody-antigen interaction. If you look to this figure, you can see the antibody interacts with antigen, and the single strength, or the strength of a single antibody in antigen interaction, called antibody affinity. That's the affinity with this one. Whereas in the strength of all interaction combined, like IgM or IgG, IgM typically has 2 affinity antigen binding site, but there are 10 of them باعتبار انه يحتوي 5 وكل antibody to Fab portion, then totally 10. The strength of all interactions called antibody avidity. Sometimes ممكن تكون للantibody اكو similarity with others when some of epitopes الابيتوبس هو البارت من اللي ممكن يتحد ويا الفابورشن when some of epitopes of an antigen are shared by another antigen, then a proportion of the antibody directed to the antigen, and this phenomenon called cross reactivity. مثل ما نعرف ان الانتيجين البارت اللي ممكن يتحد ويا الانتيبودي نسميه بالابيتوب. بعض الابيتوبز ممكن يكون إلى shares with other antigens، يعني اكو similarity between antigens، وبالتالي هذا يسمونه بالcross reactivity. What's the main function of antibody? The first function of antibody neutralization of infectivity. What's the infectivity? The neutralization of infectivity. Neutralization is the ability of antibody to bind to and inactivate فيروس infectivity، يعني المكان اللي يرتبط به ويسوي inactivation للفيروس other microbes يسمونها neutralization. If you look to this figure, we have two kinds: without antibody, with antibody. If there is infection of cell by microbe, as you know, this is the microbe, this is the cell, this is interaction, and the infected epithelial barrier, and then this is infected cell, then this is without antibody, and then the release of microbe from infected cell to another, to the adjacent cell, then release of microbe from dead cell to another cells to infected the other cells. And if the pathogenic affected of toxin, then this is the toxin and pathogen affected of toxin cell necrosis as an example. But with antigen, antibody try to block binding between microbe and other cells, because this antibody will bind with antigen and prevent binding in the cell. And also antibody blocks the infection of adjacent cells, and antibody blocks binding of toxin to cellular receptor. The other function, the phagocytosis. As you know, phagocytosis اللي نسميها بالبلعمة هي عملية engulfment. الماكروفاج تسوي engulfment للمicroorganism. As you know, this is the major step. This is the major step of phagocytosis. But antibodies, the main function of antibodies facilitate phagocytosis of foreign substance by a process called opsonization. عملية enhancement للphagocytosis. As you know, this is the opsonization of microorganism by IgG. We have antigen and then antibody binding. This is the phagocyte, has a receptor to this antigen. And then this is binding of opsonized microbe to phagocytes. This is the interaction with FCR. And then this is the engulfment. This is a phagocyte of microbes. And this macrophage try to release some enzymes other inside this one. I will describe it in the lecture of phagocytosis, and then killing of ingested microbe. The third function, antibody-dependent cellular cytotoxicity, which is called ADCC. ADCC occurs when antibody form a bridge between the infected target cell, like virus infected cell of the host, and FCR bearing effector cells, particularly natural killer cells. إذا تشوف هنا بالفيجر موجود عندنا, this is the antibody coated cells, this is the antibody, this is will bind to the surface antigen, well, that's the surface antigen, this is the antibody. Then the antibody coated cell with antibody يرتبط ويا natural killer will lead to killing the antibody coated cells, then lysis of the cells. Other complement-mediated cell or pathogen of infected cell, antibody will activated complement, بالتالي راح يسوي destroyed للبكتيريال by lysis these cells. احنا راح نعرف ان شاء الله بمحاضرة الcomplement in details. We have three kinds of complement, classical والalternative, the main two kinds of complement. هنا عندنا الانتيبودي اللي هو الاي جي ام والاي جي جي للسب كلاسز اللي هم يسوون اكتيفيشن للكومبلمنت سيستم اللي بالنتيجه راح يسوي لنا لايسز للاورجانيزم اور انفكتد سيلز. ان امبورتنت باي برودكتس راح يصير عندنا كاسكيد، سلسله من الانزيمات اللي تبدي بهذه العمليه the first recognition عندنا is C3b, which is a protein fragment that kind non-specifically to cell and antigen antibody complexes. وبالتالي ممكن يصير عندنا بعملية البايندينج تبدأ بعملية اللايسيس بنهاية الكومبلمنت procedure. Binding of antigen antibody complexes by the C3b receptor of NRBC allows it deliver the complex to the liver or spleen, where resident macrophages remove them without destroying the RBCs. Okay, we have an antigen presenting cell. I'll describe it in the complement lecture. عندنا الترانزوسايتوسيس. عندنا some antibodies can move across epithelial cell. هذا راح يكون depends on the property of constant region of that antibody molecules عن طريق process يسمونها بالترانسايتوسيس. بعض الانتيبوديز يمكن يصير كروس لايك الاي جي اي والاي جي جي. الاي جي اي اللي هو ميجر immunoglobulin ممكن يصير له حاله ترانسايتوسيس، ممكن عن طريق secretory form اللي هو بالmucosal cell of respiratory or gastrointestinal and urogenital tract. عندنا الاي جي جي ممكن يصير له كروس للبلاسنتا، وهذا واضح لكم اللي هو الوحيد اللي يصير له كروس للبلاسنتا. اوكي، بعد ما انتهينا من الستراكشر للانتي بودي وعرفنا الانتراكشن بيناتهم كيف يكون، راح نجي لموضوع الدايفرسيتي. انا ببدايه المحاضره ذكرت ان هناك billions of antigen we have 5 kinds of antibody. واحنا عندنا قاعده انه لشرط لكل انتيجين لازم يكون انتيبودي مخصص. فكيف ممكن نعالج هذه الموضوع؟ فالجنريشن للانتيبودي دايفرسيتي، اذا نجي للستراكشر مالت الانتي بودي فراح اشوف عندي multiple v region gene بالجيرم لاين، many genes coding for a variable region، وبالتالي ممكن يصير change. عندنا ممكن يصير عندي somatic recombination between elements forming the v region ايضا، like number of gene segment could recombine to give a complete v region. او ممكن يكون عندي gene conversion إذا إجانا a panel of pseudo gene can also be used to alter sequence with a variable region. هذا كل الموضوع لحد الآن يكون بالvariable region. ممكن يصير عندي إضافة للنيوكليوتايد addition during cutting and joining of the DNA extra nucleotide be inserted. ممكن يصير عندي somatic mutations also may have any mutation may be have a role to make a diversity للantibody. فالماملز may use all 5 mechanisms اللي ذكرناهم سابقا to generate diversity and other possible mechanism. إضافة عندنا ممكن للخمسة عندنا recombination inaccuracies or assorted heavy and life change. وبالتالي ممكن يصير عندنا 7 mechanisms generated diversity للantibody. In a human, the gene encoded kappa and lambda light chain are found on chromosome 2 and 20 respectively. يعني الجابا بال2 واللامدا بال22, whereas the heavy chain gene locus on chromosome 14. اوكي، عندنا الايزوتايب والايديوتايب راح نحكي عنهم. the genes for isotype variant اللي احنا قلنا بالجابا الجاما ب4 وبالتالي ممكن يعطينا 4 احتمالات، والام باحتمال، والالفا ب2 والدلتا ب1. الجابا واللامدا ايضا ممكن يكون changes. فكل هذه الميكانيزمز الخمسه اللي ذكرناهم، اضافه للاديشنال 2، اضافه للجابا واللامدا والكروموسوم اللوكس مالتهم كيف يتغير، اضافه للايزوتايب للاي جي ام 1 للاي جي 4 للالفا 2 للدلتا 1 للابسلون غيرهم. كل هذا السب جروبز ممكن يعطينا اكثر من نوع من الانتيبودي. ممكن ايضا عن طريق الاولوتايب والايديوتايبز الانتراكشن اللي راح ممكن يكون بالهيفي تشينز ممكن يصير تشينجز بالايديوتايبز. ممكن ايضا يكون التشينج اللي راح يكون بالباروتوب بالبارشن منها واللي ممكن راح يتحد ويا السي دي ار. فالايديوتايب refers to the diversity at antigen binding site اللي يسمونه بالباراتوب، المكان اللي ممكن يرتبط به الانتيبودي اللي يرتبط ويا الانتيجين يسمونه بالباراتوب، مثل ما قلنا بالإيبيتوب بالانتيجين. and in particular related to the hyper variable segment منطقة ال variable segment الhyper variable segment. These determine the binding specifically the antigen binding site the hyper variable region and sometimes referred to as a complementary determining region اللي يرمزوا لها بالCDRS. ممكن يكون عندنا جين recombination ايضا اللايت chain genes recombine ال V and J segment to make gene وبالتالي ايضا بال VL whereas ال heavy chain ممكن يصير recombined بال V وال D وال J gene segment to make gene for ال V لل variable heavy domains. ايضا ممكن يكون several VK genes on the germ line بال DNA وال JK segment and only consent بال K genes وبالتالي كل هذه ال recombination ممكن تعطينا اكثر من antibodies ايضا بالتركيبه مالتهم. Also in the heavy chain region is a variable region encoded by a V and J segment gene and the third gene اللي هو D اللي يسمونه diverse segment gene. LD is highly variable both in number and sequence of base paired. ايضا عندنا بالvariable heavy chain عندنا اكثر من واحد من ال V لل N غير محدد وال D region diversity heavy change also عندنا من 1 إلى 30 الجي من 1 إلى 16 وبالتالي راح تعطينا أكثر من احتمال more than one D segment may join to form a large D region. أيضا عندنا 87 للvariable heavy segment are found in the chromosome 14 which is at least 32 pseudogenes ممكن ينطينا وبالتالي recombinant ممكن يكون بالV والD والJ segments also. كل هذه ممكن تنطينا الدايفرسيتي بالنسبه للانتيبودي. وايضا ممكن يكون عندنا حالات السوماتيك ميوتيشنز اللي ممكن نشوفها هنا. الاي جي جي heavy and light chain genes undergo structural modifications after antigen stimulation. There is some evidence that the V region in particularly mutable the somatic mutations occur in germinal center to produce higher affinity للantibody. The high process يسمونها affinity maturation. So the potential for increased diversity multiples بعد each step of immunoglobulin productions. هذا المقدار أنه قد أكون نهاية الدايفرسيتي وعرفنا كيف ممكن يكون عندنا أكثر من antibody against antigens. إذا كان هناك سؤال يا ريت أنه تكون عندنا وقت للأسئلة وأنا مستعد للإجابة إن شاء الله. والسلام عليكم ورحمة الله تعالى وبركاته.